Welcome to the video series that is related to my efforts to develop nanomaterials against ongoing severe acute respiratory syndrome coronavirus 2 SARS-2 pandemics. Understanding SARS-2 and its pathology is essential for the success. Besides, is SARS-2 an engineered virus, and if so, what are the possible implications? What is the gravity of this situation? So will we leave the pandemics of SARS-3, SARS-4, and even more? What are the coronaviruses? Coronavirus is a protein complex not alive outside the cells. It is alive within the cells during replication and infection. Coronaviruses first described in 1962 and named in 1969. And they are circular viruses. They have a circular protein coat, and their size is 80 nanometer to 160 nanometer. The typical trait of all coronaviruses is that they have 15 nanometer club-shaped projections, also known as spikes. And this overall structure resembles a crown, and corona is crown in Latin. So this circular coat with the spikes guarding and carrying 30,000 long amino acid, long viral RNA, and this is carrying this from one host to another to continue its replication. So this is the simplified structure of coronaviruses, that it has a circular protein coat, and its diameter is 80 to 160 nanometer, and it's covered with spike proteins. Those are the spikes, and they are 15 nanometer in length. So they are interacting with the host, and this whole structure is protecting this RNA over here inside the protein coat. It's moving this from one host to another. So how they infect the human? So they first infect an intermediate animal. But in the bats, coronaviruses are digestive system viruses. So the excrements of the bats containing coronaviruses contaminate the water and other food resources, and intermediate animals like camels, pangolins infected from those sources. Especially in underdeveloped countries, then humans infected by coronaviruses adapted to these animals, and that's called zootonic transmission.